All right. Each parent has AB blood type. Great job, Xander. Good job. Okay. We are going to continue notes. So remember, your test is on Friday. I originally had your notes and syllabus statements due today. I gave you an extra day because we're going to finish them tomorrow in class. So in your Google Classroom, I did extend the due date. Your notes and syllabus statements are due tomorrow. Okay. Today, we will finish human blood types and sex-linked traits. All of the practice problems are going to be your responsibility. And we will do pedigrees tomorrow. You have 2 questions on your test about pedigrees. Okay, so we have time to get our notes mostly done. However, if we don't finish sex link today, it's going to become homework. So let's work quickly. Okay, so we left off here, right? You guys wrote in. Oh. You guys wrote this in. We talked about antigens and all of that kind of stuff, right? So we know what makes blood different from each blood type. And we know that that's because of the gene, the isoagglutinogen gene. And we know that this is because we have multiple alleles, right? We have 3 possible alleles to give us blood types. And we can have codominant or we can have regular dominant recessive. So make sure you have the table there that you know your alleles, okay? Because you're going to need that for your practice problems. I'm going to skip this video. I would highly suggest you watch it on your own time as review. There's another video as well. Mr. Anderson, he explains it very well also. Okay, do we have this practice problem in our notes? Go to your blood types. Dr. Paul? Dr. Paul, this should be number 2. His father's blood type A, his mother's blood type B, right? Yes. Part C, blood types, practice problems. If you haven't done number 2 yet, do that right now. Xander, I see your question. I'll answer that in a bit. Dr. Paul has blood type O. His father was blood type A, and his mother was blood type B. What were the genotypes of his parents? And what are the possible blood types and ratios expected for crosses involving Dr. Paul's parents' genotypes? Whew. So we got to work backwards, right? So we'll work this one over here. Again, sorry, Xander, that you can't see me, but try to hang in there with my explanation of what I'm saying. Okay, so we're working backwards. Dr. Paul, is he going to go inside the Punnett square or outside?
Inside.
In this question, is he representing the child or the parent?
Child.
The child. So it says he is blood type O. His father and mother had these other blood types. So Dr. Paul was a child. Okay. We know blood type O. What's the genotype for blood type O?
Little I, little I.
Little I, little I. Very good. So somewhere in this box, we're going to put little I, little I. You could put it anywhere. For reasons, I'm going to choose to put it down here. So you could put it in. We know we're having at least one little I possibility in the children of this Punnett square. Okay. Now, what do we know about his parents? His father was blood type A. So like we said, there's 2 ways to get A. Homozygous A or heterozygous A. So which one does his father have to be?
Heterozygous.
Heterozygous, good, because if he's little I, right, he has to get one of those little I's from his dad. So the only way to do that is to have heterozygous dad. So we'll put heterozygous dad on the outside top. Okay, so we got dad dealt with. His mother was blood type B. Okay, similarly, there's 2 ways to get B. She's going to have to be heterozygous as well. Because because he's little I. One comes from mom, one comes from dad. So she also has to be heterozygous. Very good. Okay. Now we have our parents filled out. The question continues to ask, what are the possible blood types and ratios expected for crosses of those parents? So fill in your Punnett square. And notice that it's asking us for ratios. So you have to put your answer in ratio. All right, so you're filling in and counting your ratios. So for example, it might be 1 to 1 or 1 to 2 to 1 or something like that. Okay. So we fill in our box. And is it asking us for genotype or phenotype? For the possible blood. It says, what are the possible blood types? What were the genotypes? Yes, sorry. It's asking for the genotypes of the parents. So the first question, what are the genotypes? We know that. Sorry, the second part that's asking the ratios. Is it asking for the genotypic ratio or the phenotypic ratio? Blood type. Is that genotype or phenotype? Phenotype. Because the genotype causes the phenotype, right? So the genotype would cause the overall blood type. So in this case, it would be asking us for the phenotypic ratio. Okay, well, let's count what we have here. We have how many AB? One. Okay. 1A, 1B, and 1O. So your ratio would look like this: 1 to 1 to 1 to 1. 1. Awesome. You're kicking butt. Your test has been written. Definitely have some Punnett square action on there. Okay. So make sure you do these practice problems. I will plan to post an answer key for the practice problems Thursday nights. But you should have them done before you look at the answer key. Yes? Yes. Yeah, exactly. Don't be a copier. Okay. So here is another working in. Ooh, they did percentages. Ooh, they did it wrong. I'm saying if the test asks you for ratio, you better give me a ratio, right? If it said what were the blood types and percentages, then this would be correct. But the answer still makes sense, right? 25%. All right. Andrew, are we good on this question? You got it? Looks like you got it. Perfect. Interesting facts. You don't have to write this down. Although ABO blood group antigens are regarded as red blood cell antigens, they are actually expressed on a wide variety of human tissues and are present on most epithelial and endothelial cells. So basically the antigens on the red blood cell, you can find on other cells as well. Each human red blood cell expresses about 2 million ABO blood group antigens. And functions of the ABO blood group antigens are not actually known. Like in the video, it said, we don't really know why it evolved this time, but it did. And we know that it matters because we can't do transfusions the wrong way, right? But we don't actually know why it is the way it is. All right, we have more practice problems. Are these in your? Yes, this is number 1 and number 3. Sorry, I don't know why they're all out of order. All right. Let's do them real quick. I'm going to actually give you guys a few minutes to do it. Work with your group. Write it down. Okay, show me your work. I would say pretend this is a test question. Don't just do it in your head. Write the answer on a piece of paper as if you were answering a test question. Be formal, show the work, be clear about your answer, all of that. Orlando, you said you already did these two? Okay, I would get it out. If you're stuck, ask your group. If you're still stuck, ask one of them. Ask yourself. I'm actually going to run to the bathroom real quick, you guys. I'm going to pause the recording, Xander, because we don't need to listen to just silence. So I'm just going to pause. All right. Sorry, Xander. We're recording now. We're going over the answers. Oh. Number 2, again, almost always do the first one. That's what mom was there. And pop it again. All right, mom is what kind of blood? A. If she has a child with okay, so she might have both. Okay, so thinking that he might be the father, who will this child be? Maybe. Who would this child be? C. This child would be, could be one of them, you don't know. And then this child would become A. Right? So if the dad, so he is heterozygous. So what would this mean? So can this work? So could this be the baby dad? He could be the father. Right. Okay, but why? Because right. See how yeah. So that's number 2. Thank you. Awesome. I have a random question. I have a no name on a test. Who did their test in green gel pen? Gel pen? The Unit 16 retake? Possibly. Does this look like her writing? Okay. Let's keep going. I actually want us to get to sex link. So these practice problems are in there as well. Yes, number 4 and number 5. Okay. So like I said, I will be posting an answer key for the practice problems. So you should do this on your own time first. Let's talk about sex linked traits. So you're gonna go a little backwards in your notes, right past blood type. You have a sex linked traits page. There are 2 links there that might be good to watch. You can access them in the, actually I might have them in the presentation. I probably do. Definition, here's your definition. You should be writing. Traits coded for by the genes found on the X or Y chromosome. But not both. A trait will either be X chromosome related or Y chromosome related. It will not be both. And as you guys saw on your Unit 16 test, with sex-linked traits, we see a different inheritance amount in males versus females. Specifically, X-linked traits are more common in males. Okay? That's something you definitely need to know about sex-linked traits. All right, y'all on the right page? I'll definitely be bringing tons of pictures. There's rules. I'm not allowed to bring him to class. Says the law. Okay. I'll just bring him and hide him and hope he doesn't cry. Okay. Why are most sex-linked traits on the X chromosome? Why? What would make no sense here? Because the X is bigger than Y. Sounds good to me. There are many more genes on the X chromosome, is what you're going to write down. But more genes because there's more space, right? Just as a comparison, the X chromosome is about 1,400 genes versus only 200 in the Y. Don't worry, boys, you have an X chromosome, so you don't have to miss out on all those genes. Then you are more likely to be affected. Well, you will be affected.
How do we know if we have a mess up?
Right? It like won't even erase it. Yeah, we have some. It's too good. Okay. FYI only, X and Y chromosomes do contain short regions of homology. Remember, homology means same stuff at one end. These homologous regions, the yellow in this image here, allow for the pairing synapsis of the X and Y chromosomes during prophase 1 of meiosis. This we need to watch because your next job is to describe Thomas Hunt Morgan's discovery of sex-linked genes. In your description, you should definitely be mentioning what he did with the fruit flies, all about fruit flies. Okay? So from this video, you are going to write a description. Sound good? Can we handle it? It's 5 minutes long. Turn off the lights.
While observing fruit flies, Morgan discovered a male that had white eyes instead of the typical red ones. He mated the white-eyed male with a homozygous red-eyed female. The entire F1 generation, whether male or female, had the dominant red eyes, just as Mendel would have predicted. However, the F2 generation held a surprise. Just as Mendel would have predicted, 3/4 of the offspring had red eyes, and 1/4 had white eyes. But there was a catch. Only males had white eyes. 1/2 of the males had red eyes, and the other half white eyes. If sex and eye color were being inherited independently of one another, as Mendel would have predicted, then 1/2 the white-eyed individuals should be female. Obviously, eye color was somehow linked to sex. In order to explain his results, Morgan hypothesized that while there is a gene for eye color located on the X chromosome, there is no corresponding gene on the Y chromosome. Thus, females, which receive 2 X chromosomes, also receive 2 genes for eye color, while males, which receive 1 X and 1 Y chromosome, receive only 1 gene for eye color. Looking at Punnett squares based on Morgan's hypothesis helps explain the results of his experiment. Morgan started with a white-eyed male that produced only two types of sperm. Sperm containing the X chromosome and along with it the alleles for white eyes, and Y chromosome sperm that contained no alleles for eye color. The female fruit fly, being homozygous for red eyes, produced only eggs carrying the alleles for red eyes. In the F1 generation, the female offspring are created when the X chromosome white eyes sperm unite with the X chromosome red eye eggs. Because all the females inherit one white-eyed gene and one red-eye gene, they are all heterozygous for eye color. The F1 generation males are created when Y chromosome no eye color gene sperm unite with X chromosome red eye eggs. Thus, all males have red eyes as a result of the single eye color gene they receive via the X chromosome of their female parent. Crossing the males and females of the F1 generation produces an F2 generation in which one half of the females are homozygous for red eyes, inheriting one red-eye gene from their mother's X chromosome and the other from their father's X chromosome. And one half are heterozygous, inheriting the white-eye gene from their mother and the red-eye gene from their father. One half of the males are red-eyed, inheriting a red-eye gene from the mother's X chromosome and no eye color gene from their father's Y chromosome. The other one half are white-eyed, inheriting the white-eye gene from their mother's X chromosome and no eye color gene from their father's Y chromosome. The outcome of Morgan's Punnett square is consistent with the actual breeding outcomes. The Morgan hypothesis isn't just about predicting eye color in fruit flies. It is also relevant in explaining the inheritance of a number of genetic diseases in humans. Genes that are located on one sex chromosome but not the other are referred to as sex-linked. Nearly all sex-linked genes are found on the X chromosome and are often referred to as X-linked genes. Humans carry genes related to color blindness, hemophilia, and muscular dystrophy exclusively on the X chromosome. The fact that males have only one copy of the genes for these traits puts them at a much greater risk for these illnesses than females, as they have no chance of inheriting a second good gene that will mask the expression of a bad gene they may inherit.
Am I what? No, I don't even have a Twitter. I also don't have a TikTok. And I technically do have an Instagram, but I haven't looked at it in years. I do have a Facebook. I don't even know. I've been on Reddit to read things, but I don't contribute on Reddit. Here's my description. It doesn't have to match yours, but you should be writing something. I do like the little icon. Little alien thing. Let's turn the lights back on. Okay, so from this, you definitely want to know the name. What's his name? Thomas Hunt Morgan. And you want to know that he studied fruit flies, specifically eye color in fruit flies. And he found through his breeding at one point that in the F2 generation, didn't it say only males had white eyes? Yes. So that was interesting to him. So then that led him to make further hypothesis. And through further testing, he was able to determine that the trait of eye color was on the X chromosome, the gene, I should say. So I have 4 points here. You don't have to copy mine, but okay, if you like. So basically, he's credited for like discovering sex-linked traits. Okay, so we have Mendel is the father of genetics. I would argue that Thomas Hunt Morgan would be like the father of sex-linked traits, because he was the one to discover it in fruit flies. And you should know that in sex-linked traits, males will be more likely to have the recessive trait if it's on the X chromosome. Okay. Yeah, and he saw white-eyed females. But where it was interesting to him is that when he crossed the male and female in the F2, only the males had white eyes. And that's because, yeah, it was less likely for the females because white eyes would be recessive, right? So if the females have one red and one white, they'll be red. So the females have two chances to get red. Males only have one chance. And if that one chance is white, it's white. Right? Okay, the next thing in our notes is notation. So you have here, okay, so you don't have to write this part down yet. But we should know, obviously, doing sex links, you should know where the X and Y comes from, right? And then if it's a sex linked dominant recessive trait, we're still going to have the capital A or the lowercase a. If it's a sex linked trait, the Y will have no superscript. Because if the gene is only on the X chromosome, it wouldn't make sense to write it here. So the Y indicates male with no superscript because it doesn't have that gene. And that's exactly why males are more likely to get certain diseases that are on the X chromosome. So we're still going with our capital recessive dominant, capital lowercase dominant recessive, but the Y will not have a superscript. Okay. So what you should write down, a non-diseased homozygous female. So non-diseased homozygous female, top right. XAXA, capital A, capital. We're writing this down as definition because most sex-linked genetic diseases are recessive. Any sex-linked disease that we talk about will be recessive. So you will not get any tricky questions where I flip it around and have to figure it out. So always a non-diseased homozygous will be capital, capital. We sometimes call that normal female. Second thing on your list is a heterozygous female, which we would call a carrier, right? A carrier because she does not have the disease. Yes, but she has the allele that can give a baby the disease. So she's carrying the allele in her genotype. And then the only way to get a diseased female is to be homozygous recessive. Okay? For a male, a normal male, you only have one chance to be normal. So you have to have the capital A. And remember, you should never be writing a superscript on the Y. The Y is just there as a placeholder to show that it's a male. And when we do our Punnett squares, we'll see you always get 50% girl, 50% boy. And then a diseased male, even if they only have one recessive allele, that is their only allele. So there is no dominance to cover up that recessive allele. Therefore, they will have the disease. Okay. So we have this section to go back to, and we'll do that. We got 10 minutes. Okay. Hemophilia. You guys were all like, you skipped this one. Why did you skip this one? Well, here we are. Back to this table. Hemophilia. Is it dominant or recessive? Recessive. So that's in your first box. Is it sex linked? Why? Yes. What is the effect The mutation. Oh, did I not put any red on this? I'm sorry. Nope, I didn't do any red. My bad. So, the normal hemophilia alleles prevent from making proteins that work together in blood clotting process. So basically, this second paragraph, mutated hemophilia alleles code for altered or missing proteins that cannot participate effectively in the blood clotting process. As a result, blood clots cannot form properly in response to injury. That's what I would write down. Blood clots cannot form properly in response to injury. These problems with blood clotting lead to continuous bleeding that can be difficult to control. All right. In response to injury, yes. A female with one altered copy of the gene is a carrier. Carrier females have about half the usual amount of coagulation factor. This should be in your table. So hemophilia, you can lead a pretty normal life, but it can get difficult if you have a large injury, right? If it's just a scratch, you're gonna bleed more than normal, but you can still usually stop it with pressure. It's not that your blood never clots. It just has a harder time doing it. I don't think so. I know certain drugs will thin your blood, so they can be harder. It can be harder to clot. Usually it will eventually. Just sometimes it's too late. I know my mom, one time she has really bad headaches, so she had taken one medication for her headache, which was a blood thinner. And then later she took like a Motrin or something, and she forgot that that was also a blood thinner. So what happened was in one of her eyes, all the blood vessels got really red because her blood was so thin that it was like flowing through more than it should have been. And she got nervous because she said, now I know my blood is really thin right now. She was on a long drive. And so she's like, if I get in a car accident, I could be screwed because my blood is so thin right now. So she was like really kind of scared about that. Luckily, you know, nothing happened. Nothing, and eventually her blood went back to normal. It was just because the medication that she took was thinning her blood. Because if she got a large wound, she would bleed out. Yeah. Yes.
I thought with blood thinners, you can't. It's hard to get on.
Well, it depends on what she takes for her headache is like she'll get a bad migraine and she takes like one pill. It's not a daily medication. There are other medications that might be a daily thing that are blood thinners, and that's a different thing. Yeah, and I think you would have to taper off if you can even go off at all. Depends on what you're taking it for. Red-green color blindness, also recessive, sex-linked. If it says X-linked, that means sex-linked, right? Because X is a sex chromosome. Recessive, X-linked. What happens to color blindness? You're colorblind. Let's see. Mutations in the genes associated with the color blindness cause affected individuals to have trouble distinguishing between some grades of red, yellow, and green. These changes lead to an absence of cone cells or to the production of abnormal opsin pigments. I think we can just stop at the basics here. I do not think at the level that we are learning at, you have to know like all the specifics of red, green color blindness. You just need to know. Yeah. No. Yeah. Yep. And if you haven't heard, they do have those glasses that can correct for some kinds of color blindness now. There was some famous YouTuber guy that made like a video about it. I forget who it was. But you put them on and then everything's like more normal. My brother-in-law has color blindness and he got the glasses and he said it definitely made a difference, but he was just so used to his life before that he would just rather keep living that life rather than get used to normal life. So he actually doesn't use them very much. All right, we have 5 minutes. Let's figure out what we need to do. Again, video, great video. You should watch it. We have practice problems. I think we should try to do one really quick. Hemophilia is a recessive sex-linked trait caused by a defective. Is this an actual problem? What now? Number 1 for part. Horrible. I'm sorry, but I have to write on this side. Okay. Okay, number 1, part D. So, the normal allele produces a protein called factor 8 that allows blood to clot. Without injections of this factor, hemophiliacs are at risk of dying due to excessive bleeding. Make a Punnett square to show a cross between normal male and a female who is heterozygous. So, normal male. What? First, we have to start with what to show a male? X, Y. And this allele is only on the X chromosome. So we're not going to write any superscript on Y. And if he is normal, he has to have what? Capital or lowercase? Capital. We're going to go H of hemophilia. And what are we crossing? A female who is heterozygous. So we show XX for female.
So capital, lowercase.
Capital, lowercase, because heterozygous. Wonderful. At this point, I know the term, what was it? Plug and chug? I used to plug and chug. Plug and chug. Okay. Fill it. We need to come up with a phrase for Punnett squares. Plug and Punnett. All right, so we have, and what's the question asking us? Expected genotypic and phenotypic outcomes. We have 2 sets of answers. So let's go one thing at a time. X big H, X big H. How many of those do we have? 1. X big H, Y. 1, 1. Okay. So our answers, what are we doing? Ratio or percent? Outcomes. Okay, so I'm going to do 25% XH, XH, big H, big H. 25% X, big H, Y. 25% X, big H, X, little h. And 25% X, little h, Y. Okay. Now with phenotypic outcomes with sex linked, there's actually kind of a few ways you could do it. You could just do normal and disease, or you could split it up into normal female, disease female, normal male, disease male, carrier female. I generally like to go more specific, okay? With IB, unless it says be general, you should probably be specific. So we have 25%. What's big H, X, big H, X, big H?
Hetero? No, homo.
Normal. Normal who?
Oh, normal female.
Normal female. She is not a carrier. She does not have the disease. She is normal. Okay? Let's do our other. Well, let's do in this order. We have 25%. What's X, big H, Y?
Normal male.
Normal male. Very good. We have X, big H, little h.
Carrier.
Very good. Carrier female. And 25% disease male. Okay. I like to say affected. Because saying you're diseased kind of sounds But I guess that technically is correct. You could say that. I like to say affected. Affectedness. Okay. Looking at your packet, I know it's time. I know, I totally know. There is a little section on Punnett squares. So I'm gonna give you that for homework. Okay, it's not, guys, it's like not that much. Slides 106. I'll send you a reminder. You might want to write it down. What page of the notes is this? I promise I'll have your next note packet numbered. So this is page. No. I'm sorry. Oh my gosh. Okay, page 1, 2, 3, 4, 5, 6, 7, 8, 9. Page 11. You're gonna do page 11, which is slides 106 to. And this is easy. It's gonna tell you how to fill in a Punnett square. You guys all know how to do that. But look at this fun gift. Okay, you have slide 106 to 118. So if you're writing down your homework, you need to do page 11, slides 106 to 118. Are we good?
Yes.
I'll send you a reminder. I'll see you guys tomorrow. We will finish our notes tomorrow. You will have your test on Friday. Oh, we're going to be absent Friday. So you're going to take your test without us. You can do it. I believe in you. Yeah, we're going to Disneyland, so you guys have fun. No, I'm kidding. We're going to be at the Esplanade site doing a training. So don't worry, we'll be suffering along with you.
Like, do you want to go? I'm like, no, I have class.
Oh, I would definitely say yes.
Yeah, you're not a student.
No, you're not a student. Well, I'm glad that you have that amount of Disneyland. Yeah. I'm like a Disneyland fanatic. All right, Xander, are you still there? I'm going to pause the recording.